Hello everyone, and welcome to a special edition of the ActinoBase e-seminars. So this week, lots of us in the Actinomycete research field should have been in Toronto for the ISBA conference. Sadly, we can't be there for obvious reasons. So we thought we'd mark the occasion this week with a special ISBA session of the ActinoBase seminars. So shortly we'll be having a few flash talks from early career researchers in the field. But to start the session off, we're going to have a short introduction from Justin Nodwell from the University of Toronto, who's kindly agreed to speak to us today to give a little history about the ISBA conference. So I will just hand over to Justin now.
Thanks very much, Alicia. Let me just share my screen here. And there, can you see that? Okay. Let's see. Oops, I don't know how to advance my slides. There we go. Okay, so Sam emailed me a couple of months ago and asked me to give a short talk about ISBA, the history of ISBA, to mark the occasion when we were supposed to be meeting, which I thought was a terrific idea, and I agreed to do it immediately. And then as soon as I clicked send on the email, I had a sort of a sinking feeling when I realized that I actually don't know all that much about the history of ISBA. In fact, for most of my career, I've been referring to it as the International Society for the Biology of Actinomycetes, when it is in fact the International Symposium on the Biology of Actinomycetes. Gilles van Wezel, a colleague in the Netherlands, corrected me on that about a year and a half ago, which strikingly was 27 or so years into my career in this field. Anyway, I emailed a whole bunch of people, asked them for their stories, and had great fun gathering information on about ISBA back through the years. But in the end, I decided that rather than talk about that, I would just talk about, give a little bit of what I've learned about the history of ISBA. And mostly I would talk about why we need ISBA and trying to put that in the context of this field as I understand it. So without further ado, I'll move on to that. So the field of the actinomycetes started in the 19th century with this guy. This is Ferdinand Kohn. He was a very important microbiologist in Germany. And in 1875, he described an organism that was an actinomycete. He named it Streptothrix forsteriori. He also described it as a thing called a ray fungus. So it sort of the field started off in a state of confusion. Is this a prokaryote or a eukaryote? And furthermore, he isolated this organism from a human tear duct, which it occurred to me that as these organisms can be rather uncooperative as experimental subjects, the fact that the first discovery of it was in the context of something that made people cry might resonate with some of the graduate students who are listening in. The next step forward in this field came with a fellow named C.O. Hartz, who I've been unable to learn anything more about him. And he described a microbe called Actinomyces bovis, which caused, he isolated from the pus from an infected jaw in, I believe, a cow. So this field that we work in started with a fairly unauspicious beginning. We started with confusion as to whether we're looking at a bacterium or a fungus. Is it prokaryote or eukaryote? And furthermore, it was something that caused infections in certain barnyard animals. So inauspicious start, but things went uphill from there. Several decades later, we get to this guy, Selman Waxman, who discovered a molecule called streptomycin, which is produced by the bacterium Streptomyces griseus. He showed that this molecule has potent antibacterial activity and developed it as one of the first widely used natural product antibiotics. It's arguably one of the most important drugs in history. This work went on to kick off certainly the modern antibiotics field with the discovery of also of tetracycline and some of the aminoglycosides and other compounds. You could argue as well that it really kicked off the modern drug discovery field in many ways as well. So this is absolutely seminal work in the history of medicine, and he was awarded the Nobel Prize for this work. And if you fast forward a couple more decades still, you get to this guy, Satoshi Omura, who, and there's an excellent legend about this discovery. He discovered the compound avermectin, which is produced by a bacterium called Streptomyces avermitilis, which apparently, I'm told that he isolated from a soil sample from a golf course not too far from where his laboratory is located. This sort of lore is widespread in this field. People who work on actinomycetes find these microbes in the strangest of places, from caves to the bottom of the sea to the tops of mountains. Anyway, he was able to show that avermectin, he subsequently developed into a semi-synthetic called ivermectin, had potent activity against nematodes and insects, and it was developed as the first successful, widely used anti-parasitic drug. It's worth noting that in an act of extraordinary generosity on Omura's part and also his collaborators in the private sector, this drug was made available to countries in sub-Saharan Africa, which had challenged healthcare systems and a lack of disposable income to pay for expensive drugs. It was made available at cost or in many cases for free. And as a result of this, the disease known as river blindness was virtually eliminated in the space of something like 5 years. It's an extraordinary accomplishment. And he was also awarded the Nobel Prize for this work, I believe in 2000 and actually not sure when it was, but he's a second Nobel laureate in this field. So that's a pretty remarkable foundation for what we do. I think if you want to fall in love with an actinomycete, really all you have to do is streak it out on a plate and watch it grow. This is a plate of Streptomyces silicolar that was struck out in my lab by one of my first two graduate students. It was either Tamara O'Connor or Michael Hudson. I'm not sure which. But this plate for me really, this is why I first fell in love with this organism. It's just the sheer beauty of this organism and the number of visual cues that goes along with its life cycle. You can see that there's this, this layer of white fuzz. I don't know if you can see my cursor. Anyway, there's a layer of white fuzz on the colonies. That's the spore-forming cells. And there's also this beautiful blue pigment being secreted into the medium. It's an antibiotic called actinorodin that's been studied by many labs, most famously by David Hopwood's lab in the UK, but many others, including my own lab, we've studied it to some extent as well. Watching all these things unfold in real time is a real treat. You start off with a colony that looks like a little beige blob on a plate, and then over the next couple of days, it erupts into all these wild activities that are really striking to watch. This is driven home, I think, by this image here. This is the cover of the 1991 abstracts book from ISBA 1991, which was held in Madison, Wisconsin. This image is attributed to somebody named Jerry Ensign, I believe. And I think it really drives home the remarkable complexity and weirdness of these bacteria. You can, well, I can just zoom in a little bit. You can see that there are many of these organisms have what are filamentous cells. So that's one major feature of the actinomycetes, except that some of them, like the mycobacterium, do not. You can see that these bacteria are often have branching cells in most cases, except in others, like the arthropacteria, where they do not. You can see that they have sporulation mechanisms that sometimes make their spores in these lovely sort of whimsical little curlicue structures here, these beautiful chains of spores that you find in Streptomyces silicolar. And then other organisms like some of the Nocardia or some of the other Streptomycetes like Venezuela rather, make their spores in these ramrod straight filamentous cells. Still others make their spores in things that look like little meatballs, like the actinoplanetes here, or in other things that look like big bunches of grapes, like the Micromonospora. Still others, like some of the actinoplanetes, make spores that can swim. Really fascinating story has developed over the past, I don't know, half decade from the Onishi lab on that story. So these organisms, this whole cluster of microorganisms is really characterized by something I would say is morphological anarchy. The molecular biology is fascinating. The cell structures is fascinating. The life cycles are fascinating. They're really just breathtakingly amazing organisms, but also very confusing and somewhat difficult to study. I would argue that confusion, or let's say contradiction and paradox, is one of the underlying core principles of this field. So this is a page from an early paper in the field. This is from the Hopwood lab from 1965. And this describes work that Hopwood was doing genetic linkage mapping. So it's possible to mate different strains of Streptomyces silicola together and look at the segregation of oxytrophic markers like proline oxytrophy, histidine, or arginine oxytrophy in the progeny, and thereby map the distances between the various genes that are involved. So pro, his, and arg here. And in that way, what they found was that the genetic map of Streptomyces silicola is circular. And I've actually done these kind of experiments before. It's true. This genetic map of Streptomyces silicola is a circle, and that suggests that the chromosome is a circular molecule. And that was the accepted truth in the field for some time. However, as time went by, certain things started to not add up. And so about almost 30 years later, Kanashi and Murayama were able to show that the giant plasmid SCP1, which is also found in Streptomyces silicola, is not a circle. It is in fact a linear molecule. And a few years after that, David Hopwood, working with Carton Chen, Helen Kieser, and Yixing Lin, showed that in fact the Streptomyces lividans chromosome, Streptomyces lividans is very closely related to Streptomyces silicola. They're able to show that this chromosome is also linear. It's a linear molecule, which was, I remember when this paper came out, this is just, I had just started my postdoc, and this was a real blockbuster, and I turned the field on its head. It was Joe McCormick was the first person who told me about this, and it was quite something. The story continued to unfold, and so a group in France, Bernard Descaries, with Pierre Leblanc and Gilles Fischer, were able to show that the end regions of Streptomycete chromosomes, they're working in ambiphations, but this applies to all the Streptomycetes, have these gigantic inverted repeats, hundreds of kilobases long, that have all kinds of implications for the relative stability or instability of these chromosomes. And I think there's many questions that remain to be answered about why they are like this and what it means for Streptomyces growth and replication. And finally, there's a discovery by Kai Bao and Stanley Cohen that in fact, these giant linear plasmids and linear chromosomes, the very ends of them are characterized by a repeated DNA structure sequence that has a proteinaceous factor associated with it, I believe covalently, that essentially functions as a telomere. So this tells you a lot about this field. We start off with an organism isolated from tear ducts that is maybe a fungus, maybe it's a bacterium. Some years later, we discovered that these microbes make an extraordinary diversity of biologically active small molecules, some of which can be harnessed as antibiotics or antifungals, some as antiparasitics, some as anti-cancer drugs, immune suppressants, and also a myriad number of really interesting and useful chemical probes that can be used to study everything from cell wall biogenesis in bacteria to RNA trafficking in eukaryotic cells. It's really just an extraordinary degree of diversity to this field. So I think that tells you quite a lot about this field. It is fundamentally, these organisms are fundamentally, there's just something anarchic about them. Anarchic and maybe a little chaotic. But in the end, they're just bacteria. So what does this have to do with ISBA, and why do we need ISBA so much? I've been going to conferences for a long time. And I've been a scientist for a long time too. And perhaps you have an experience that I've had many times, which is that I have friends who are not scientists and who I try to explain my work to, and I can see them. I can almost check my watch and calculate how long it's going to be before they start to fall asleep. For the layperson, it's just hard to follow what we do. So sometimes I'll just tell them about conferences I'm going to. And when you tell someone you're going to, say, the general meeting of the American Society of Microbiology, and it's in Washington, DC, they usually understand immediately, well, that sounds very important. You should go there. That's good. But if you tell them that you're going to the International Society, there I did it again, the International Symposium on the Biology of Actinomycetes, the effect is not quite as good. It has a sort of an anesthetic effect on non-scientists. So then I will sometimes tell them, well, the conference is in Turkey this year. And then they'll sort of slap their forehead and will have their view confirmed that my job mostly consists of me not working and going to exotic locations for conferences where I hang out on beautiful beaches and talk to my friends over drinks. There's a tiny bit of truth to that. In any event, I think the world doesn't understand actinomycetes. I think even most scientists don't understand how exciting actinomycetes are. And the only people who really do is us. And so we constitute a society of sorts, even if we don't have a bricks and mortar address. ISBA, I think, is the only meeting where the extraordinary diversity, complexity, and fascinating biology associated with these microbes is captured at all. And so this is why we need it. And there's several other things about this meeting that I truly love. As I say, I've been to lots of meetings, Gordon conferences, ASM, European conferences, everything. But I always come back to ISBA just because it's so great. And I've learned a lot about this from my colleague, Gilles van Wezel, who's the director of ISBA. I'm just running the conference. And one of the things I learned from him was that this meeting is inherently international. And it has been, I followed up with a couple of other colleagues, Keith Chater, David Hopwood, and Mike Goodfellow, and they confirmed that really was intentional and goes back to the very origins of this meeting. So if you look at the list of locations where this conference has been held, the very first meeting was held in Germany, specifically in East Germany in 1968. I think for the younger people who are listening, it's hard to appreciate the significance of that. This is 1968 is the height of the Cold War. The division between East Europe and West Europe was a profound one. There was a brick wall with soldiers all along it, separating those two blocks. And yet, this tiny group of scientists met in crossed borders and met and shared ideas about science. And in fact, there are no less than 3 meetings behind the Iron Curtain prior to 1989. So there's the German in 1968, there's Poland in 1974, and there was Hungary in 1985. And you can also see that the conference has been held at myriad locations. And this is really appropriate. When I joined this field, the acknowledged leadership in this was pretty much in the United Kingdom and Japan, I would say, with honorable mentions going to Germany and a couple of other countries. But if you look back at the authors of those papers I talked about, and some of the other papers as well, you can see that there have been major contributions to the actinomycetes field from virtually every country on Earth. There are major contributions from China, Korea, Japan, Mexico, from South American labs, Italy, Spain, Canada, the United States, Taiwan, you name it. The many people, a tiny number of people from the entire world have contributed to this field over the years. So we're a remarkable community. And this is built into the genetics of this conference, if you like. The Gilles impressed upon me that every effort is made to ensure that there are speakers from every continent and every country, if possible, where actinomycetes research is going on. It creates certain challenges. Sometimes you're listening to a seminar or having a conversation with somebody who you don't really quite share the same language, and you're limping along. But it's a really worthwhile endeavor. My impression is that maybe the conference hasn't done quite so well on balancing gender of speakers, but that's something we're working on, and it improves steadily, and we'll try and do well in Toronto when we gather in Toronto. Another thing I love about this conference is that it tends to take on something of the local character of where it's held. So some of you no doubt remember the closing ceremony at the Jeju, Korea conference in 2017, which is just spectacular, this Korean traditional dance thing. It was really great. But my favorite example of that, and I have to plead a certain amount of national chauvinism, is this. This is the emblem from the 2001 ISBA conference. This is actually the first ISBA that I attended. So this was held at the University of British Columbia. It's a beautiful university in the middle of a beautiful city, Vancouver. And this symbol you can see behind it, it's interesting that there's no, the symbolism that has been chosen behind this emblem is indigenous. So this is a very ancient symbol of many of the Pacific Northwest indigenous nations. This one I believe is Haida. It's an extremely ancient symbol, certainly thousands of years old. And it represents, and if there's any indigenous people listening, you must accept my apologies. I don't know as much about this stuff as I should. I believe this represents the thunderbird. The thunderbird is the most powerful of gods to these people. And it's a god that can change its shape. It's a god that can control the weather, and it can change into the shape of a person and influence human behavior. And I think in many ways, so it's kind of a god of chaos and a god of order, which I think is very appropriate to the whole study of Actinomycetes biology. But this emblem also tells another story, which I suspect was lost on most of the people who attended that conference. But I'll share it with you now. The University of British Columbia is located on what in Canada we refer to as unceded territory. There's no treaty or deed that says that the indigenous people of that land ever gave it up. And in fact, the land where UBC is located is the traditional grounds of the Musqueam people of that region. They have lived there for, it's believed, tens of thousands of years. It's true of many universities in Canada. It's true of my own university, University of Toronto, which is located on unceded land of the Seneca, the Mississauga, and the Huron-Wendat people. So this is what I love about this, is that in addition to teaching each other about science, we have lots of other things that we can teach each other as well. You know, going forward, I suspect that a lot of scientific conferences will move to this kind of forum where we're speaking to each other online. But I hope that some meetings survive as person-to-person conferences. It's worth it, and it's important for us to actually be in the same room sometimes. So on that note, I will end, and I'll just say thanks very much for listening. If COVID cooperates, which we're hoping it will, we're going to be holding ISBA in 2021 again in Toronto. I've been in conversations with Felix van Wezel about dates for this, and we're hoping to have a set of dates for you very soon. It will be almost the same structure as it usually is. It will start on a Sunday and end on a Thursday, most likely. And if you want to watch for this, I'll be announcing the dates on my Twitter, and I'll reach out to ActinoBase and to also get more publicity, and I'll also send it out through the ISBA mailing list. So on that note, I'll thank you very much for listening. And if there are any questions, I'd be happy to take them, but otherwise I'd be very happy to just turn this over to the students. So thank you very much.
Amazing, thank you. That was really interesting. Yeah, so as Justin said, if you have any questions, you can reach out on Twitter. So now we're going to move on to our flash presentation from some early career microbiologists. So first up, we have Katie Noble, who is a PhD student at the University of East Anglia. So Katie, if you want to share your screen and get going. And if you have any questions for the flash presenters, we're probably going to only have time for one or two, but if you post them in the chat, we'll try to get them.
Give me a second, sorry.
No problem.
Okay, right, so hi, as Sam said, I'm Katie. I'm a first year PhD student in the Hutchins lab at the University of East Anglia in Norwich. And I'm going to be talking a bit about what I've been doing for the first 8 or 9 months of my PhD, looking at the regulation of this novel antibiotic, formicomycin, in Streptomyces formica. So, as everyone knows, antimicrobial resistance is an ever-increasing problem with the emergence of superbugs and resistant strains that can't be treated with the antibiotics we currently have available in the clinic. So, this timeline here shows some key aspects of how this crisis has been able to develop. Just comparing the top and bottom lines, you've got the antibiotics when they were deployed and then how quickly resistance has been observed. So you can see in quite a lot of antibiotics, it's quite soon before resistance can be seen, and in some cases, even before an antibiotic has been deployed on a large-scale clinical level. There's also an issue with some of the dates. So you can see in the 1940s and 50s, there was a large surge in new products being identified, particularly from the natural products of actinomycetes, but this was followed by quite a distinct void, as repeated discovery of known strains in the natural products meant that there was nothing new being identified. But advances in sequencing technology and genome editing tools has brought actinomycetes back to the forefront of discovery. The strain I've been looking at is Streptomyces formica, or KY5, which is isolated from the nests of Tetrapnera benzigi ants that colonize these swollen thorns of acacia plants in Kenya. So a couple of years ago now, the Hutchins lab sequenced the genome of KY5 and identified 34, but potentially up to 45 biosynthetic gene clusters, making this quite a talented strain. So bioassays showed it to have activity against MRSA, which unfortunately I don't have a picture of, Bacillus, Candida, and Lamentaspora. So it's got quite a broad range of activity against gram-positive and negative bacteria, as well as some fungal pathogens. So responsible for some of this activity, mainly the antibacterial activity, are these three distinct groups of compounds here, the phasomycins, formicomycins in the middle, and then formicopyridines. These are produced by cluster number 26, which encodes a type 2 polyketide synthase, and these are all have a pentacyclic ring structure with slight differences in the R groups and the closed and open structure in the phasomycins and formicomycins, and then a pyridine moiety in the top left of the formicopyridines. So the phasomycins are precursors to the formicomycins, and then the formicopyridines are produced as shunt metabolites when that pathway gets disrupted. So you can see here, there's quite a nice story to my project of what the problems are and then how KY5 could be part of the solution. These are the genes involved in the biosynthesis of these compounds, so I'm not going to go into a great amount of detail, but you can largely see the biosynthetic genes are in the red. You've got the core PKS ones in the middle here. Transport genes are in the blue, 4AA and 4K. Those are mainly just involved in export of the secondary metabolite once it's been produced. And then the regulators are in green, and that's what I am interested in. So we've got 4J and 4Z on either end here. They're both MAR regulators, but they have relatively low sequence identity to one another, suggesting that they carry out quite different roles in the regulation of formicomycin. And then 4GF in the middle is a two-component system. So Dr. Rebecca Devine in the lab has spent all of her PhD project and now her postdoc looking at this whole strain and specifically the formicomycin cluster, and made CRISPR knockouts of each of these to identify what their roles are. So when 4J is removed, formicomycin production increases around 5-fold, deeming it the main repressor of the pathway. When 4GF is knocked out, formicomycin production is completely abolished, deeming it the main activator. And when 4Z is knocked out, formicomycin production reduces to around 65% of that in the wild type, suggesting that it's an activator, but not necessarily essential in the whole pathway. Rebecca has also done ChIP-seq on these to identify the binding sites of these three regulators, and this is where my project has been following on from. So I've been looking at the effect of that binding on promoter activity using gut assays. This uses a beta-glucuronidase gene in front of your promoter of interest and adding PMPG to the sample. If the promoter is active, this enzyme will be produced and can hydrolyze PMPG into PMP, which is a lovely yellow color you can measure with a spectrophotometer as a quantitative indicator of promoter activity. So I haven't got any time today, unfortunately, to go into a lot of detail about the results, but hopefully there will be a paper being published soon with all of the data in it. So yeah, these are my contact details if you are interested in finding out more, and thank you to everyone in the lab and all of the organisers for everything today.
Awesome, thank you for that, Katie. So we have time for one or two quick questions. If anyone has any questions for Katie at all. I've not seen anything yet. We'll just give it a couple of seconds. Maybe.
That's fine. I'm happy to answer things on Twitter if anyone has any questions later.
Yeah, that's fine. And if anyone posts anything in the chat, we can circle back as well.
Yeah, no worries. Thank you very much.
Okay, cool. Awesome. Thanks for that, Katie. So next up, we have Linamaría Pintor-Escobar, who's a PhD student at Hill University in the UK. So, Lina, if you're happy to share your screen, I will.
Yeah. Okay, hi everybody. My name is Lina. I'm a second-year PhD student at Lorena Fernandez-Martinez group at Hill University. And my research focuses on 3 isolates from a high altitude ecosystem called Páramos in the northern Andes of South America. And this place has a quite unique set of climatic conditions, and it is considered also one of the main evolutionary hotspots in the world. However, their microbial diversity remains underexplored, and there is a great opportunity for drug discovery. So a few years ago, some soil samples were taken from one of these páramos in Colombia, and 3 isolates from there had or show a quite broad range of bioactivities. So my PhD focuses on characterizing these 3 strains in their potential for antibiotic production. So with these strains, we started by sequencing their genomes. Then we wanted to understand the taxonomic status of these isolates, and to achieve a good resolution for species delineation, I use a multi-locus sequence analysis with 5 housekeeping genes. And the results show that the strains 885 and 893 are the same species, and they can be classified as Streptomyces pratensis. Also, the results show that the strain 926 is part of this well-supported clade, but when we look at the evolutionary distances, it seems distant enough from these other strains. So we suggest this strain is a new species of the genus Streptomyces. So with this in mind, we move into doing some bioassays, and this time we target pathogens of clinical relevance. And here I'm showing you just the results for E. coli and Klebsiella pneumoniae. But as you can see, the strain 926 show a very strong inhibition halos, and it looks very promising. So we prepare some extracts, and in collaboration with Barry Wilkinson's group at the Junior Center, we run some chemical analysis. So when we look at the chromatograms from the HPLC, we found a set of peaks that didn't appear in the blank. And it seems that these peaks are all compounds from the same biosynthetic pathway. And then when we did high resolution mass spectrometry, we found out that one of these peaks seems to be a compound, rare halogenated compound with at least 1 chlorine atom. So when we went back to genomes, and we looked for a cluster which could encode a compound of those characteristics. And we found these 150 kilobases cluster, which has NRPase genes and PKase genes. And it has also 2 halogenases. So it seems like this could be the cluster responsible for that molecule. What I want to do for the rest of my PhD is to find more characteristics of this molecule that might be responsible for the antimicrobial activity we see. And I intend also to confirm the link between this cluster and this molecule. And by understanding the role of the genes involved in this cluster, I aim to improve the production of this compound. So that's it. Thank you very much, and I'm happy to take any questions.
Awesome, thanks for that, Lena. Yeah, we'll just give this a couple of seconds to see if any questions filter through. But again, if you post them in the chat, we can potentially circle back to them at the end of this time, if you haven't thought of any yet.
Okay, there is a question.
Yeah, shall I read that out? So the question is, why is Páramos such an evolutionary hotspot?
Yes, well, I spoke about the very unique climatic conditions. So these places are located very close to the equator. They happen to be only in a few countries. I think it's Colombia, Venezuela, Ecuador, and Peru. And because of their location and their high altitude, they have high rates of UV and also very cold temperatures. But there is still vegetation. And I think all these combination of climatic conditions make these places one of these evolutionary hotspots. They are under so much selective pressure that they evolve very, very fast.
Oh, cool. Interesting. So there's one more question we can get to. This question is, does your novel CG926 isolate show any special morphological characteristics?
Well, it has a beautiful pigment. It is it's like a pink pigment, and that's beautiful, but we haven't done a full morphological characterization of this strain. We haven't looked at the spores in SEM, or we haven't done other type of analysis, so I can't really say about that.
Okay, cool. Interesting. Well, thank you for that. So our last flash presentation for today is from Dr. Divya Prajapati. So you can just switch to the camera, mic, and share your screen. She's based at the Maharaja Sayajirao University of Baroda in India. So when you're ready. Sorry, you'll need to just turn on your microphone and camera, if that's right again.
Hello.
Hello, yeah, ready when you are.
Yes. Hello. Am I audible?
Yeah, we can hear you. So whenever you're ready.
Okay, fine. Hello, everyone. Welcome to my presentation on the topic indispensable role of adiabatic chromomycin in physiology and metabolism of the producer organism Streptomyces flaviscleroticus. I did my PhD from Biochemistry Department, MS University, Baroda, India. I started the most exciting and thrilling journey of my PhD with the Streptomyces, a very special organism, which is antibiotics are considered as secondary metabolites because they are produced at the tail end of the growth of the producer organism. So my thesis work, it focuses on my thesis work focuses on challenge that it is challenging the present study focuses on the challenges that the conventional wisdom that the role of antibiotic production is to confer fitness advantage merely by inhibiting the growth of other organisms. The organism of my thesis is Streptomyces flaviscleroticus, which produces antibiotic chromomycin, which is an organic class of antitumor compound and aromatic polypeptide in nature synthesized by type 2 TCS cluster. The chromomycin deletion mutant was constructed in which 12 kb DNA containing minimal CKS genes were targeted for deletion, and expected phenotype when we delete any antibiotic cluster is just loss of antibiotic production. But along with these expected phenotypes, we observe certain unexpected pleiotropic phenotypes, like the mutant was reduced growth of mutant, the chromosome deletion mutant JP1. Then it lost the sporulation, and also there is lack of growth on a minimal media containing glucose as carbon source. Moreover, the JP1 mutant loses viability very early in the stationary phase, as we can see from this confocal image of live dead imaging. Now, it was questioned that that whether the unexpected phenotypes we are getting are because due to some secondary mutation. So we inserted back the 12 kb DNA back into the chromosome of JP1 mutant, and we got integrant which was indistinguishable from the wild type, and thus ruling out the possibility of any secondary mutation because of which this phenotypes are we are getting this phenotype. So my thesis work focuses on the unusual phenotypes. So first of all, as we see earlier, that the mutant loses viability very early in the stationary phase. So first of all, when we consider viability of lifespan of any organism, it is intimately linked with the better management of oxidative stress. If the organism doesn't manages properly oxidative stress, then it may lose viability very early. So we tested various biomarkers of oxidative stress. Among those biomarkers, I'll be explaining some where we see there is a sudden rise in catalase activity at the very early log phase, and which decreases in stationary phase, while in the case of wild type and integrant, the catalase activity rises during stationary phase. Moreover, the extracellular H2O2 production is also high in JP1 as compared to wild type and integrant, and the DCFDA probe, which is used to detect reactive oxygen species inside the cell, it was also high in the case of JP1 as compared to wild type and integrant, and thus concluding that JP1, thus we concluded that the JP1 mutant is under constant constitutive oxidative stress. So this constitutive stress led to metabolic rewiring in JP1. As I have already explained that the JP1 mutant fails to grow on minimal media containing glucose as sole carbon source. The main reason is because of downregulation of glycolysis. The first committed step of glycolysis, the PFK, was analyzed, and we found activity almost 2 to 3-fold low in the case of JP1 as compared to wild type and integrant. And the same thing was observed at expression level, but I have not shown the expression level over here because of space constraint. Moreover, we found upregulation of pentose phosphate pathway, and here I have shown activity of glucose 6-phosphate dehydrogenase, and we have monitored few other enzyme activities and also expression analysis. The metabolic rewiring is occurring in order to augment NADPH production for reducing power generation, and we definitely see here that in the JP1, the reducing NADPH levels are higher as compared to wild type. So the combined effect of metabolic rewiring and constitutive oxidative stress led mutant unable to grow on minimal media. The inability of mutant to grow on minimal media can be rescued by supplementing amino acid at 0.5% concentration, which is much higher than the oxidotrophic requirement of any organism. So at 0.5% concentration of amino acid, the JP1 mutant growth can be rescued on minimal media. Moreover, in minimal media, if we supplement the amino acid, which are get degraded to alpha ketoglutarate of PCA cycle, it can also rescue the growth of JP1 on minimal media, but the rest of the 15 amino acids can't help in the rescue of the growth of JP1 on minimal media. And since there is downregulation of glycolysis, upregulation of PPP, there is definitely impairment of PCA cycle enzyme to contain oxidative stress, and hence the alpha Kg levels are also low in the case of JP1 as compared to wild type. Moreover, because of this whole rewiring, we found the acetyl-CoA levels also very much down in the case of JP1 as compared to wild type. So we thought that why there are so many pleiotropic phenotypes we observe just by deleting antibiotic chromomycin. We tested in vitro antioxidant property of chromomycin, and indeed chromomycin scored very much better in all the in vitro antioxidant assays as compared to polypeptide yellow color antibiotic tetracycline, and we use ascorbic acid as control over here. And further, we tested whether the in vitro antioxidant property of chromomycin do have any in vivo repercussions. So we supplemented the subinhibitory concentration of chromomycin at the start of the growth of chromomycin deletion mutant JP1, and then we saw dramatic results. First of all, the chromomycin at subinhibitory concentration significantly enhanced viability of JP1 mutant, as we can see from confocal images over here. The JP1 mutant, which is losing viability in stationary phase, can be a significant enhancement in viability, we can see. And the same thing can be seen in the plating experiment, which I have not shown over here. Moreover, the ascorbic acid, which is proven antioxidant, can do the same job. And thus it proves that, yes, indeed, in vivo experiments, the chromomycin is acting as antidote at subinhibitory concentration in the JP1, also reduces the intercellular levels of reactive oxidation species, extracellular H2O2 production, and also the NADPH levels are maintained in JP1 if the chromomycin is provided from outside. Moreover, it also reverses the loss of activity of PFK catalase as well as expression and TCA cycle enzymes, as we can see over here. So in conclusion, the ex vivo supplementation of chromomycin rescues majority of the defects of the chromomycin deletion mutant, but it can't reverse the growth of defect of JP1 on minimal media with glucose as sole carbon source. And the probable explanation for this could be inherent preferences as compared to glucose as carbon source. Then there is a reconfiguration of carbon metabolism due to constitutive oxidative stress in JP1. And secondly, inability of exogenous chromomycin provided during the growth to mimic its intercellular regulated production. Because of these three reasons, we think that the chromomycin supplementation didn't reverse the growth of JP1 on minimal media containing glucose as sole carbon source. Now, we see that all these effects which we see are because are at the growth related growth phase effect of chromomycin. But we already know that this it's believed that the chromomycin production is temporarily segregated in stationary phase. So we did expression analysis of the chromomycin biosynthetic gene cluster at the start of the growth, and really we found that the chromomycin biosynthetic genes are expressed as early in the midlog phase, and thus providing the clue that yes, the expression of genes of chromomycin early in the growth phase of the wild type, the expression of chromomycin and production of chromomycin helps in physiology and metabolism of the producer organism. Thus, this whole story provides a new perspective that the secondary role of secondary metabolite chromomycin in the physiological and metabolic effect of the producer organism. And the importance of this study could be, this role could be attributed to those cryptic gene clusters which are not generally expressed under normal conditions. So they might have some functions like to the producer organism. Otherwise, why would an organism harbor such a complex genes for such a complex molecule and apparently gain no advantage from it? The work that I did during my PhD is published in Environmental Microbiology, really online library. Thank you very much for listening to me talk.
Great stuff. Thank you for that. So if there's any questions, you can post them in the chat now. I'll just wait a couple of moments to see if anyone has anything to ask you. But obviously, if not, people can reach out to you on Twitter, I assume. We tagged you, I think, in one of the posts from ActinoBase. So Yeah, okay. If anyone has any questions for Divya, feel free to reach out on, oh, actually, sorry, we do have one from Justin here. So Justin is asking, is there any indication that this happens with the other redox active antibiotics? The effect?
Yeah, there is one study for phenazines. The phenazines, they are proved it is redox active antibiotic, and they have physiological role in Pseudomonas aeruginosa. And Newman's lab at Caltech, they are doing great work on this.
Cool.
For Streptomyces, it is not done. For Streptomyces, it's not done so far. But for other organisms, for Pseudomonas, for Myxovaricin, Myxovaricin is an antibiotic produced by Myxococcus xanthus. If Myxovaricin is deleted, then its predatory effect is lost. So basically, for these two organisms, it is reported. But for Streptomyces, this is for the first time we are reporting the direct benefit of the antibiotic to the producer organism.
Cool, interesting. So Justin just had a small follow-up about actinorodin. I guess no one's tested it, right?
Yes.
With actinorodin, if anyone's, I presume no one's. Yeah, no, it's fine. He's just a little, but yeah, he was just mentioning actinorodin anyway. But thank you for that. That was really interesting. Thank you to all of the presenters and to Justin for his great talk at the beginning. So we are slightly changing the format from now. Instead of doing these weekly, we're going to start doing them every other week, just because some of us are trying to get back to work. So we have a bit less time to organize these sessions, unfortunately. So we'll be back on the 9th of July, I believe. Yeah, we're back on the 9th of July. We will have, I believe that will be another one of the longer talks from a group leader. So again, just to say again, we are still accepting talks from early career researchers or from group leaders. So if you'd like to give kind of a 40-minute overview of the work your lab do, we would love to hear from you, and you can sign up on the form. If you'd like to give maybe a 20-minute talk about your postdoc or your PhD research again, we'd also love to hear from you, so you can sign up via the form. And we may do another one of these flash presentations in the future, so feel free to sign up for those still as well. And if we get enough interest, we could definitely do another one of these sessions. I think they're really, really great. So thanks everyone, that's all for now. Cool, that's the stream over.